Hey Team Bio, welcome to your screencast, a deep dive into transcription and translation. So I know we've already talked about this in the process of talking about organelles and introduction to cytomembrane cycling in the cell. We talked about the central dogma and this idea that genetic information flows from DNA to RNA to proteins, and proteins lead to your traits and behaviors. And all that information is encoded within the DNA. And we're about to embark on a unit in genetics where we talk about how DNA is inherited and then manifested as traits and behaviors. And so I think it's important to spend a little bit more time just talking more explicitly about what's happening in transcription and translation, because really this is the secret of life. Understanding how DNA encodes information and then how it's turned into an action item, a protein, has profound implications for human ability to manipulate the natural world. I don't know if I can impress upon you enough the importance of understanding this pathway and how much power that gives a person. Okay, anyway, off my soapbox. Let's get right to it. So, transcription begins in the nucleus with the DNA. It is the process of taking DNA and making RNA. So this blue double helix is our DNA molecule, and this red ribbon is our RNA. So notice what's happening here. The DNA is opening up. Remember, we have complementary base pairing. Within the DNA, A always binds to T and C always binds to G. So the base pairs are split down the middle, and then an enzyme called RNA polymerase scoots down the DNA in this direction, and it brings in a complementary base pair to the DNA. So I know this looks like a red ribbon over down here, but know that it's actually composed of individual nucleotides of RNA. So the next nucleotide is going to come in here. It's going to have complementary base pairing between the DNA, and it will add on like this. And then there'll be another RNA added on here, also with complementary base pairing with the DNA on next. So this ribbon is going to grow in this direction until it reaches the termination sequence when the RNA polymerase is going to fall off. So this is our growing, immature mRNA. It's called an mRNA because it's a messenger RNA. It's coding a genetic message. Okay, so the RNA is going to continue to grow until the RNA polymerase reaches the termination sequence, in which case it's going to fall off, and then we're left with a long strand of mRNA. The mRNA is immature, and there's something called, a few things have to happen. It needs to have a 5' Mg cap, methylguanine cap put on. It needs a 3' poly A tail. Tail. And it needs to undergo splicing. So what are all of these things? The 5' methylguanine cap, you can see right here, is just a cap that protects Mg cap. Is shown here in yellow, and the poly A tail is a bunch of nucleotides adenosine put at the end here. And Splicing is when a piece of the mRNA message is cut out. So see all these kind of light red portions of the mRNA? These are called introns. And confusingly, introns are cut out of the message. They're called introns because they stay in the nucleus. The pieces that are going to be part of the final mRNA message are called exons. So anything that's a dark red here is an exon, and anything that is a light pink is an intron and is going to be cut out of the message. So these exons will be, it's called spliced together. So the message will look like this at the end. The exons will be joined back together, and the introns are cut out and remain in the nucleus. So when that has happened, when the methylguanine cap has been added, the poly-A tail has been added, and the splicing has been completed, we now have a mature mRNA, which is going to leave the nucleus through a nuclear pore and end up in the cytoplasm. Okay, and this is a nuclear pore as well. So you can see here the mature message is leaving the nucleus, and it's going to end up in the cytoplasm, where it's going to meet up with a ribosome. This is a part of a ribosome. So a ribosome is composed of two subunits, the large subunit and the small subunit. And the two subunits join together, I spelled that wrong, on the mature mRNA message, and they are going to scan along the message until they reach what's called the start codon. So know that this looks like a ribbon, but it's actually got teeth on it. There are nucleotides, the nitrogenous bases sticking out from one side. So the ribosome is going to scan along the message. It's going to run in this direction until it reaches the start codon. And what happens next is illustrated by this process, and I'm going to talk to you about it in a second. But first, I'm going to go up here and tell you what these little weird candlestick things are. This is called a tRNA. It stands for transfer RNA. A tRNA it looks like a candlestick here, but it actually takes on a cruciform shape, kind of like a cross. And it is just like the mRNA. I know it doesn't look like it. It's a folded strand of RNA composed of a bunch of nucleotides of RNA. And it's got one special kind of action end, which is called the anticodon. The anticodon is composed of 3 nucleotides in a sequence. And the anticodon that any tRNA has specifies the amino acid that's going to bind on this end. This is the amino acid binding site. So in the cytoplasm, we have tRNAs that are free, and they're going to be charged up with a specific amino acid. All these purple dots, or different shapes, specify different amino acids. So a tRNA, as you can see here, is going to be charged up with a specific amino acid. And then we'll talk to you about this next process. So the next thing, we have already gone from DNA to RNA, and that was transcription, and now we're going for translation. And this is the process of taking an RNA message and making a protein. Okay, so the ribosome has 3 sites: the arrival site, the polymerization site, and the exit site. The sequence of events is such. A tRNA a new tRNA comes into the A site, the arrival site. Why are you doing that? Sorry, guys. I don't know why it's not letting me. Okay. A tRNA arrives at the ribosome in the arrival site. It has, as I said before, an anticodon. The anticodon is a sequence of 3 nucleotides that binds to a codon, which is a sequence of 3 nucleotides, in the mRNA message. This base pairing is specific. A always binds to U, and G always binds to C, and vice versa. So the message is specific. If this is an AUG here, it means that the anticodon must be UAC. And tRNA with the anticodon of UAC is always bringing a specific amino acid, which is actually a methionine, to the party. So every set of 3 nucleotides in the mRNA is a codon, and which binds to a specific anticodon in the tRNA, which is bringing a specific amino acid to the party. Okay, so the anticodon and the codon are going to bind together. And then this tRNA is going to be directly next to this tRNA, which is holding a polypeptide chain. Now, when this tRNA is securely in the A site, and this one is in the P site, there's going to be a polymerization event where this amino acid is going to break the bond here. This bond is going to be broken between its tRNA, and there's going to be a new bond formed to this amino acid here. So this purple square is now going to be directly attached to this purple hexagon with a peptide bond. Just like these two are connected with a peptide bond, and these two are connected with a peptide bond. And this will effectively transfer this polypeptide chain to the top of this tRNA. And this tRNA, which used to be holding the chain, is now free and is free to leave in the exit site, which you see this tRNA here is doing right now. So the whole chain will be transferred to this tRNA, and the ribosome will shift down 1 codon. So this guy will now be in the P site. The A site will be empty and ready for the next codon to be read and a new tRNA to come in. And this guy will be in the E site ready to exit the ribosome. So this is the secret of life. This is how we read an mRNA message and we turn it into a polypeptide, which, remember, when the polypeptide is folded, it will become a working protein. And the protein will catalyze a reaction or serve a structural function, but it will do the business of helping the cell stay alive. Now, I hope that you can imagine the implications for changing a nucleotide within the DNA. If you change a single nucleotide within the DNA, now there's going to be potentially a new, or there will be a new RNA nucleotide that is complementary to that new DNA base. That will change the mRNA sequence. If that is part of an exon, it will be part of the mRNA message that leaves the nucleus. And it will change the nucleotide that is part of the message. So maybe instead of an A here, this is a G. If there's a G here instead of an A, this means that the tRNA that comes in here will be a CAC, which will change the amino acid that is brought to the chain, which will then change the structure of the folded protein and potentially ruin its function, maybe, or enhance its function. This mutation will have an effect on what the amino acid is and eventually affect the protein. So, yeah, that is the secret of life. That is how we take information from the DNA and we turn it into a protein. I also want you to know that this code is universal. So a fragment of DNA that's in a bacteria could be put in a human cell, or vice versa. A human gene could be put into a bacteria, and then a human protein can be produced by that bacteria, by the bacterial ribosome machinery, and then it can be harvested and used because the genetic code is universal across all living things, which is pretty amazing. So you could take a gene from a mouse and put it in a human, or a gene from a jellyfish and put it in a mouse, and yeah, that's what's going on. Okay, that's all. I will see you all in class next week.